Well, we've learned about general function of cells. We've learned how cells make proteins. Now we're going to take some time and learn about how it is that cells make copies of themselves and how cells grow and eventually die. And we're going to be looking at two main processes during this set of lectures. The cell cycle, which is the series of events that occur during the lifetime of a cell. And then we're also going to be looking at something called mitosis, which is the series of events that occur when a cell divides to create two new daughter cells. Okay, so let's start by talking about the cell cycle. The cell cycle is a series of repeating events, and they're very predictable. All cells go through them. Just like we as humans have a predictable life cycle. We all know people start out as an embryo and then they become a fetus, and then they're an infant and a toddler and a youngster and a teenager and adult, and then they're an elderly person, and then they die. A cell has a similar series or sequence of events that are very predictable that occur, and the cell can go through these multiple times, or a cell may only go through it once, depending on the type of cell we have. There are 5 different stages to the cell cycle. There is the interphase phase, which consists of 3 subphases, and then there's the M phase, which consists of 2 subphases for a total of 5. Okay, let's take a look first at interphase and see what happens during interphase. Let's see, did I do that? Yep, okay. During interphase, there are 3 different, discrete times in the cell. And during interphase G1 phase is where we get normal growth. Now, if you imagine, if I had a cell and it divides in 2 to make 2 daughter cells, those 2 daughter cells are basically exactly half the size of the original parent cell. So the first thing that they need to do is to grow to adult size. They need to replace organelles that they didn't get in the division of all the materials of the cell. They need to generate some more cytoplasm. They need to basically grow to normal size again, to adult size. That's what happens during the G1 phase. You get normal growth of the cell, and it occurs right at the end of cellular division, from the last round of cellular division. This is usually a fairly long period of time. For some cells, they never exit out of the G1 phase. They just stay in the G1 phase forever. In fact, we say they become arrested, and we say that they're in a G0 phase, meaning they just never get beyond this first growth phase. Okay, so the whole point here is to recover from the last mitotic division, to replace parts that you weren't given during the cellular division. That's what G1 is about. Now, at the juncture between the G1 phase and the next phase, which is called the S phase, there's something called a growth checkpoint, or the G1 checkpoint. And this is when this cell will actually communicate with neighboring cells to determine whether or not it would be appropriate for it to undergo mitosis or not. If the environment is really crowded, or there aren't very many nutrients around, then the likelihood is that it wouldn't be a very smart idea for this cell to undergo all of the energy expenditure required to go through cellular division, in which case it would not progress on to the S phase. However, if at the G1 checkpoint the cell gets sort of the green light from all the neighboring cells, it appears that there's plenty of nutrients, there's plenty of space for new cells to be generated, there's a need for new cells to be generated. If we get the green light, then that cell will progress from G1 into the S phase. And the S phase is a very long phase. It's actually longer than is depicted here in this graph. The S phase it takes a considerable amount of time, because this is when I synthesize new DNA. In other words, I copy the existing DNA so that I have enough DNA to give each daughter cell a full complement of DNA. So I'm replicating all of the chromatids or chromosomes. Then the next thing that's going to happen is the G2 phase. And I don't know why they drew the G2 phase so large in this graph, because it's not. The G2 phase is actually very short. It's a short little period of time during which the cell synthesizes proteins that are specifically are proteins that are only used during cell division and no other time during the life cycle of a cell. So during G2, we quickly manufacture those few key proteins we need to undergo mitosis, and then we begin the events that follow that help us to divide up the chromosomal material and the cytoplasmic contents of the cell. So let me go back here. We've just finished, we talked about interphase and its three parts, G1, S, and G2. What we're going to be talking about in the next part of the lecture is something called the M phase. M stands for mitosis. And the M phase consists of a mitosis and a cytokinesis part. So we're going to be looking at that in part 2.